Today we're going to start talking about our first metabolic pathway, which of course is glycolysis. And before we get into the details of the pathway, I want to kind of set the stage by giving you some context. The biggest context I could come up with is planet Earth. And so I thought we'd see how it is that glycolysis fits into the biogeochemical cycle for carbon, which of course humans are actively in the process of disrupting by mining geological carbon that would normally stay under the ground for tens of millions of years in a geological cycle, and bringing it all up to the surface and burning it, putting some of it in the atmosphere where it will persist for some thousands of years and warm the surface of the Earth in the process. So we're kind of messing with this badly right now. But light, so the interesting thing about global cycles is that, you know, it's like an area of earth science, right? It's like geology, atmospheric science, you know, rocks and water and stuff like that. But the cycling of the elements, all the major elements in the biosphere, is actually dependent on life. So Earth's climate depends on protein structure because the photosynthetic reaction center, which takes light and uses it to fix carbon, both to make ATP by mechanisms we'll see, and to fix carbon from the atmosphere, from CO2 gas to CO2 sugar as a dissolved constituent of the cell, you know, based on light, right? That's the Earth's biogeochemical cycle for carbon, right? And if you are the kind of cell that eats reduced carbon, then you're also the kind and burns it in oxygen, then you're the kind of cell that puts out CO2, and so you replenish the CO2 that the photosynthetic autotroph took from the air, right? So there's a cycle, it's normally there, and as I mentioned, we're now messing with it by digging up fossil carbon and burning it a lot faster than geology would put it there. So these photosynthetic autotrophs fix carbon into sugar, CH2O, right? And also evolve O2, okay, as you see. Right? And then that's all for the benefit of heterotrophs like us, right, who in the process of respiration put out H2O and CO2 gas. Right? And so these heterotrophs, most of them, are engaging in aerobic respiration. Aerobic meaning oxygen. So they're burning the O2 with their fuel. And the fuel was produced because the plants used the energy of the sun to put it there. Right? So, you know, ultimately everything's going to have to come from biomass, right? I mean, when we finally run out of all the fossil fuel, we have to make stuff like plastic and everything, all the other commodities that we're used to, like enormous numbers of industrial commodities are made from fossil fuels now. We have to make it all from plants. If you're looking for a good field to go into, kind of like in applied science, this would be a great area to think about. So, you know, this is the global carbon cycle on the surface of the Earth. Aerobic respiration. And aerobic respiration, we could break down into at least 4 parts. One is glycolysis, which we're going to start talking about today. And glycolysis is actually anaerobic. Glycolysis is a very old pathway. It goes back to near at least the beginnings of life. And it certainly was around as far as any of the molecular dating technologies that we use. Certainly around before O2 started accumulating, which was on the order of 2.7 to 2.4 billion years ago. Actually, what happened is the O2 started accumulating, and then a lot of it got used to oxidize iron in the rocks. And so you have all this red, rusted iron all over the Earth in rusted formations. And after the amount of iron on the surface was taken up by this evolving oxygen, then the O2 can start to build up in the atmosphere and of course, it was originally a poison. In fact, it's still a poison, and we'll see examples of how it's a poison and how our cells now have to use mechanisms to detox because of all this poisonous oxygen around us. I mean, we need it to live, and it's poisonous. Generates free radicals, etc. Right? So other steps include something called the pyruvate dehydrogenase complex. I'm just going to write PDC because it will be the subject of most of the lecture next week. The Krebs cycle, also known as the tricarboxylic acid cycle, TCA. Right? And something we mentioned last time, oxidative phosphorylation, by which we make ATP. Right? So those 4 steps. Right? If we're talking about sugars, now if we're talking about fats, then we're going to feed that in as well. Right? So fats will feed in here in step 3. And then they'll use the same step 4, as we'll see. So that's the global carbon cycle. Any questions about that? We're just kind of setting the big picture here. If we look inside of a cell and we ask how does glycolysis fit into kind of the big picture of metabolism, we could say, well, we've got reactions in cells that are build-up reactions, right? Anabolic reactions are synthetic reactions that make macromolecules. Right? So macromolecules. And they get made from things like amino acids, sugars, fatty acids, and nitrogenous bases. Right? I mean, these are the 4 components of the cell, right? That you learned about on day 1 of the last term. And this is coupled, of course. All cells have both of these things going on at the same time, which is where regulation becomes so important, which is why we spend time thinking about how enzymes are regulated, because the biological regulation, which can be different in different tissues, comes down to regulation of enzymes, because the enzymes are running metabolism or making it go fast enough, allowing it to go fast enough so that the cell can grow and divide on reasonable timescales and therefore gain adaptive advantages. So these things have to happen at the same time. And catabolism is the breakdown. So here we have carbohydrates, and fats for the most part. To some extent, when cells get near starvation, usually they start to break down their proteins for fuel. But typically, it's usually a pretty bad situation if the cell is using more than token amounts of proteins to feed into fuel cycles, into making ATP. Because proteins, of course, are stuff of the cell in structural ways too. And so this is going to generate carbon dioxide, water, and ammonia. Okay. And so between these 2 parts of the cell, right, we're going to look at pathways on both sides, we have these sort of special factors in the middle. And we already know what they are. So for example, FAD, right, and FADH2 are in a cycle. Right? Because we're breaking down energy-rich carbohydrates to their smaller constituents, ultimately CO2, right? The carbohydrates are getting oxidized, and that means that something else can get reduced. So FAD is an oxidized form of that cofactor. We looked at it last class. FADH2 is the reduced form. So FAD to FADH2, sugar to CO2, that's a redox couple, right? There's no direct enzyme, of course, in the cell that does that. It's mediated through long, complicated pathways. But as the big picture goes, this is what's going on. Right? So similarly, we could say we have NAD or ADP, right, going to NADH or pH. Right? Same deal, just a different kind of redox cofactor. Right? And then we could say we have ATP going to ADP plus phosphate. So ATP is burned or hydrolyzed or used in some way to drive the otherwise unfavorable synthesis of macromolecules. And when we have fuel that we're breaking down, we can take the products of that ATP synthesis, ATP breakdown, and make more ATP out of it. That's unfavorable on its own, but we're driving it in coupled reactions because we have this energy-rich fuel, right, as we will see today. So these guys and these guys, these would be the energy-rich forms of these key cofactors, and these guys up here are the energy-poor forms of the cofactors. Right? We don't necessarily think of FAD versus FADH2 as energy-poor versus energy-rich, but that is kind of what it comes down to, because FADH2 can deliver its reducing equivalents, its electrons, down an electron transport chain to ultimately allow for ATP to get made. So it's connected to being able to make ATP. So I think it's not unreasonable to think about FADH2 and NADH and NADPH as three energy-rich cofactors, and they're all the reduced forms. Right? So reduced forms of cofactors are energy-rich because they can get used in oxidative phosphorylation. Right? So these guys here, electron transfer, ATP synthesis. And recall that last time we also distinguished that kind of ATP synthesis from the substrate level ATP synthesis, where you have some enzyme and ATP is getting made right on the enzyme. And today, hopefully before the end of class, but if not, then next time we will see two examples of ATP getting made in the second part of glycolysis. Right? Questions?